Hello there everyone. In today's lab we will be doing the gram stain technique. The gram stain technique is one of the most important techniques in medical microbiology, which differentiate between gram-positive and gram-negative bacteria. As you know, gram-positive bacteria has a thick peptidoglycan layer, which retains the crystal violet and appears purple under the microscope. And gram-negative bacteria has a thin peptidoglycan layer and an outer membrane. So when we stain the gram-negative bacteria, because of its thin peptidoglycan layer, when we added crystal violet and iodine, after decolorization, the crystal violet and iodine removes and safranin remains, which appears pink or red under the microscope. The material here we have the crystal violet dye, gram iodine, decolorizer, which is a 95% ethanol, and gram safranin. And we have the Bunsen burner, the slides, inoculated lobe, everything is ready. Before we do the gram staining, we need to prepare the smear. Now we are going to prepare the smear for gram. Okay, now we are going to prepare the smear. As we said before gram staining, we have to prepare the smear. First, we are going to take a clean slide and then a drop of distilled water. After that, we are going to sterilize the inoculated lobe. And cool it down. We can cool it down somewhere away from the colonies. Okay. After that, we are going to take a colony from the culture and smear it onto the grain slide. Okay, after the smear has dried, we are going to do the fixation 3 times over the Bunsen burner, and then we place the slide on the racking stain. After that, as we said before, we are going to add it crystal violet first, and then gram iodine, decolorized, which is a 95% ethanol, and safranin. First, we're going to add it crystal violet, which stays for 1 minute. After 1 minute, we are going to wash it with tap water. And then we are going to add the gram iodine safranin, which provide a violet crystal iodine complex. It also stays for 1 minute. Then we're going to wash it as well with tap water or distilled water, and we're going to decolorize it with 95% ethanol, which remains 20 to 30 seconds. Decolorized, which as we said before, the gram-positive bacteria, which contains a sick peptidoglycan layer and negative gram-negative bacteria, which has a syn peptidoglycan layer. It will remove all the crystal violet and remains the safranin at end. I'm going to wash it again with still water. And then the last step is adding safranin dye, which if it's a gram-negative bacteria, it retains the safranin and appears purple under the microscope. It stays for 1 minute, and then we observe it under the microscope with the oil immersion. We're going to wash safranin, and after that we're going to dry it with filter papers and then observe it under the microscope.